Okay, so welcome to this next video in which we're discussing the pathogenesis of rheumatoid arthritis. Okay, so we've now discussed the structure of a synovial joint and the structure of the synovial membrane. Now what we're going to discuss is the pathogenesis of rheumatoid arthritis, which is going to be a chronic inflammatory disease of the synovium, which is eventually, going to lead to the degradation of the bones that are involved in the synovial joint, and it specifically affects synovial joints of the distal extremities such as the hands and the feet, worst of all. Okay, right, so let's begin then, the pathology of rheumatoid arthritis. So it begins years and years, well, it's speculated to begin years and years before any sort of joint symptoms actually arise, okay? So there is a prearticular phase, okay? So prearticular means pre-joint, basically. So the full portion which occurs long before the portion that involves the joints. Okay, so the prearticular phase. So what happens in the prearticular phase? Well, basically, you lose immune tolerance, okay? So I should say that actually, rheumatoid arthritis is an autoimmune disease, okay? It is a attack by the body's own immune system on proteins that the human body makes itself. Okay, so the immune system is supposed to attack proteins that are made by dangerous cells, proteins that no human cell would ever make, basically, if it was healthy. Okay, so dead giveaways, basically, that something is wrong. That's what the immune system is supposed to attack. In rheumatoid arthritis, it is going to attack proteins which are made by human cells, basically. Now, the specific proteins that are thought to be attacked in rheumatoid arthritis are citrullinated proteins. And I should say that we don't know the specific proteins that is attacked. What we think, however, is that they are citrullinated proteins. So let me explain what a citrullinated protein is. Okay, citrullinated protein. Okay, so basically, you know that proteins are made up of polypeptides, okay? So polypeptides folded into some weird and wonderful shape, okay? And polypeptides, if we look at them now, imagine it linear rather than in its folded structure. Polypeptides are just a sequence of amino acids, basically. It's a polymer of amino acid after amino acid after amino acid after amino acid, etc. Okay, now, one of the amino acids that is used in proteins is an amino acid known as arginine. Basically, there is a related amino acid that is not used in proteins, which is called citrulline, okay? And you can convert arginine into citrulline, basically. And when you do this, you have to do it to the protein that's been made. You can't convert arginine to citrulline and expect the citrulline to be incorporated into the protein. It doesn't work like that. Instead, what you have to do is you have to make the protein with arginine in, and then it will be converted to citrulline, basically, once it's in the polypeptide, basically. So once the arginine is within the protein, you can convert it to citrulline, okay? And this conversion is done by an enzyme known as peptidyl arginine, peptidyl arginine, and that's all one word. So peptidyl arginine, so arginine which is in the peptide, and then it's deaminase. And peptidyl arginine deaminase is often abbreviated to PAD. Okay, PAD, peptidyl arginine deaminase. Okay, right. So let me show you the conversion. Let me show you the structure of arginine and the structure of citrulline, and then you'll understand how the two are very similar to one another. Okay, so Let's draw the basic amino acid structure for arginine firstly. Okay, so here's the core amino acid structure. So here's the amino group up here, here is the alpha carbon, and here down here is the carboxylic acid group. Okay, now the R group of arginine is that you firstly have 3 methylene groups, and because I'm lazy I'm going to use, and also I don't have much space, I'm going to use a little trick where you just draw 1, bracket it, and then put a 3 down there, which means copy out 3 of these. Okay, then you have a nitrogen with a hydrogen coming off it, a carbon here with a double bond up here to a nitrogen which has a hydrogen off, and then an amino group down here. Okay, so this is the structure of the amino acid arginine, and this is a proteinergic amino acid, so it's one which is actually incorporated into proteins. Now citrulline is very related in structure to arginine, but it is not a proteinergic amino acid, it's not incorporated into proteins. Okay, so again I'll copy out the core amino acid structure. So here is the carboxylic acid group down here, the amino group up here, and the alpha carbon in the center. And the R group of citrulline is almost identical. So again, you have 3 methylene groups, so I'll draw 1, bracket it, and put a 3 there. Then you have a nitrogen coming off here with a hydrogen off it, and then you have a carbon, and this time a double bond to an oxygen rather than a nitrogen up there, and then an amino group off here. So this now is the structure of citrulline. Okay, so this enzyme, peptidyl arginine deaminase, and it should be noted that there isn't just one peptidyl arginine deaminase, there's a whole family of them. This enzyme can convert arginine to citrulline, basically. It can carry out this conversion here. Right, and as I say, you make the polypeptide with arginine in, and then this enzyme will act on the protein and convert some of the arginines to citrulline, and when you do this, the protein is then called a citrullineated protein. Okay, now this happens, okay? The problem is that in people with who are going to develop rheumatoid arthritis, which will happen later, what seems to happen is that there is a break in immunological tolerance to these citrullineated proteins. So usually the immune system should tolerate these proteins, i.e. it should not launch an immune response against citrullineated proteins. They are a normal component of the human body. Okay, you have them. But in rheumatoid arthritis, what happens is you generate autoantibodies against citrullineated proteins. Okay, so let's go over the page and talk about this. So, what happens is that there is a break in immunological tolerance for these citrullineated proteins, and you create what are known as autoantibodies. So this means an antibody which is directed against a protein which is present within the human body, a self protein. So auto means self, autonomy means self-governing, autoantibody means self-antibodies, antibodies which are directed against proteins which are within the human body, okay, which are made by your own cells. Now, let me just draw a little picture of an antibody. Okay, so the normal antibodies, remember antibodies, or immunoglobulins as they're also called, consist of 2 heavy chains and 2 light chains. So here, these are the heavy chains, okay, and I'll highlight them in, and they're connected to one another by disulfide bonds, which I've drawn there. Okay, so here, ooh, this is smudging nicely. These are the heavy chains in red here. Okay, so these are called, strictly speaking, their full name is the immunoglobulin heavy chain. Okay, so immunoglobulin heavy chain. Okay, and then each immunoglobulin heavy chain then has an immunoglobulin light chain, which I'll show in orange here. So in orange, next to the immunoglobulin heavy chains, you have immunoglobulin light chains, which are also linked to the immunoglobulin heavy chains by disulfide bonds. So this is an immunoglobulin light chain. Right, okay, so basically for each antibody molecule you have this region up here which is specific to that antibody, okay? So let me just divide this structure up further, basically. So, basically, if we take one of these immunoglobulin heavy chains out, okay, you can separate it up into two portions. You can separate it up into a constant portion, okay, which is down here, and then you can separate it up into this other portion up here, which is the variable portion. Okay, you can do this also for the light chain. So here's the light chain, and it can be divided into a variable portion and a constant portion. Now, basically, your body, your immune system, can produce a huge number of different antibodies, okay? And this portion that's made up of these two variable portions here, which is this portion up here, this is known as the FAB portion, which stands for fragment antigen binding, and the portion which is made up of the constant portion of the immunoglobulin heavy chain and the constant portion of the two immunoglobulin light chains, which is this portion downwards here. Okay, this is known as the FC portion, which is the fragment crystallizable. Okay, so basically your immune system is capable of producing antibodies which target a huge number of different proteins. Okay, so you can produce antibodies that target protein, let's say protein A, which has then a totally different structure from protein B. Okay, so let's say we've got protein A here, which is a square protein, and then we've got protein B over here, which is triangle protein. Okay, so you can produce antibodies which will bind to protein A and antibodies which will bind to protein B. Okay, and these are going to have totally different binding sites, basically, for the antigen. And it is this portion up here which binds to the antigen. So you have two antigen binding sites, basically. And an antigen is just any protein which can have an antibody made against it. Okay, so this portion is variable between the two different types of antibody which will bind to these two different proteins, basically. Okay, so different antibodies which are targeted to different proteins will have different variable regions, whereas they might have the same constant region, although I will just discuss in a moment that constant region is a bit misleading because it can change as well, but I'll talk about that in a moment. Okay, so also what I must say is that the antibody, so if I draw the antibody here, it will bind to a specific portion of protein B. It won't bind to the whole thing. So it has a specific portion of antigen B that it binds to, and this specific portion of the antigen which the antibody binds to is what's known as the epitope. Okay, so each antibody has an epitope on the antigen which it binds to. Now, antibodies which bind to different epitopes on antigen B, i.e. you could have an antibody which binds over here, that will have to have also a different fragment antigen binding, basically. It will have to have a different binding site here, because both binding sites, this binding site here will not bind necessarily to this portion of the antigen B, it's specific to this portion. So, not only do you produce different antibodies to different antigens, but you also produce different antibodies to different epitopes of the same antigen. Okay, so you can produce a huge number of different antibodies, and they vary in their FAB region, basically, the portion that binds to the antigen. And it's because these portions of the heavy and the light chain, the variable portions of the immunoglobulin heavy chain and the variable portion of the immunoglobulin light chain, those can vary between different antibodies, and therefore you can produce different binding sites for the antigen. Okay, however, the constant region can also vary between different antibodies. However, it's less variable than the variable region. Okay, so there are different constant regions that you can use, but there are only 9 different constant regions that you can use, whereas the variable regions, you can produce hundreds of thousands of different variable regions. So there are 9 different constant regions that you can use, basically. And I should mention that this is the constant region of the immunoglobulin heavy chain we're talking about. The constant region of the immunoglobulin light chain truly is constant, but for the immunoglobulin heavy chain, the constant region can vary. Okay, and there are 9 different constant regions for the immunoglobulin heavy chain. So there is the constant mu region, there is the constant delta region, there is the constant epsilon region, there is the constant alpha 1 region, there is the constant alpha 2 region, there is the constant gamma 1, constant gamma 2, constant gamma 3, and constant gamma 4 region. So there are these 9 different constant regions. Now, if you use the constant mu region, you end up with an antibody which is referred to as an immunoglobulin M molecule. Okay, so IgM. Ig stands for immunoglobulin, and then you put M, which is the Latin analogous letter to mu, which is the Greek. Okay, constant delta, you end up with an IgD antibody molecule. Constant epsilon, you end up with an IgE molecule. Okay, and then these ones, if you use the constant alpha 1 region, your molecule is strictly speaking called an IgA1. If you use the constant alpha 2, your molecule is strictly speaking called IgA2. But people will commonly just refer to those collectively as IgA, immunoglobulin A molecules. Okay. Similarly, there are these 4 different constant gamma regions. Okay. And they would, strictly speaking, be called IgG1, IgG2, IgG3, IgG4, etc. But they're usually just referred to as IgG collectively. Okay, so these are collectively all referred to as IgG. Okay, right. So now that we've discussed the structure of antibodies, then. So antibodies have this FAB region, this fragment which binds the antigen. So FAB, remember, stands for fragment antigen binding, which is this portion which actually binds to some epitope of an antigen. Okay, so now what's going to happen is we are going to produce antibodies against these citrullinated proteins, okay? And these are going to be autoantibodies because the antigen they bind to is a self-protein. Okay, right. So, how does this occur? Well, basically, you're going to get a humoral adaptive immune response, which is going to lead to the activation of B cells, which are then going to differentiate into plasma cells and produce these autoantibodies. Okay, so something goes wrong in immunological tolerance. This should not happen. Okay, and it's likely that it occurs at the level of the thymus potentially, or maybe even at the level of the bone marrow, because there are supposed to be mechanisms for stopping the production of B cells and T cells, which are directed against antigens, against self-antigens, against auto-antigens as they're called. So antigens that are in your own body, basically. Something goes wrong. Now it's also worth recognizing that maybe it's a fault on the MHC class 2 molecule's part, because what has been found is that a certain MHC class 2 molecule known as HLA DR4, okay, and let me just explain this. So remember, MHC class 2, okay, this is the protein complex on which you present antigen fragments. So when you need to activate a T cell, what needs to happen is that a professional antigen presenting cell needs to present the antigen fragment which you want to produce an immune response against on MHC class 2 here to some naive T cell, okay, and needs to present it with co-stimulatory molecules. Now, there are many different MHC molecules, Specifically, there are many different MHC class 2 molecules. Okay, so there are 3 main types of MHC class 2. There is HLA-DR, okay, there is HLA-DP, and there is also HLA-DQ, and these are all 3 separate types of MHC class 2. So you have 3 separate types of MHC class 2, and for each of these 3 types there are many different alleles. Now a specific allele of HLA-D3 is HLA-DR is HLA-DR4. Okay, and if you have this specific allele of HLA-DR, then you can present, well, you are 4 times more likely to get rheumatoid arthritis, basically. And it is potentially because this form of MHC class 2 will present these autoantigens to T cells, and the other forms maybe are less good at presenting these autoantigens. They won't bind these autoantigens because all of these different forms of MHC class 2 and all of their different alleles, they all have different selection criteria for which antigen fragments they can present. So let me just discuss this in a little bit more detail. So basically, if you remember, MHC class 2, it's got a very large peptide binding groove. Okay, so it's open-ended. So let's say the peptide binds along here. So it can bind a peptide between 13 and 25 amino acids. And basically, in order for a peptide to bind to the MHC class 2, what has to happen is it has to have certain amino acids at certain positions. Okay, so there are set positions where you need certain amino acids because these amino acids are involved in binding to a specific portion of the MHC class 2 molecule. So for instance, here there might be some specific binding site for a specific amino acid, and here also there might be another little binding site, and here also. Okay, so these sites where you need specific amino acids because only that amino acid will be able to bind to this site, these are what determine which peptides can bind in the peptide binding groove. So you have to have a certain combination of peptides in these positions, and it doesn't matter what amino acids you have in the middle, basically. And these amino acids that you have to have at certain positions are what are known as anchor residues. Now, MHC class 2 molecules are specialized so that the combination of anchor residues that you need at specific positions, that would be a combination that you wouldn't usually have in self proteins. At least that's the attempt. It doesn't work completely, but it's an attempt, basically, that basically you have this combination. Let's say in this case we have 4 anchor residues. So let's say, just to make it up, maybe you need a certain amino acid at position 4, a set amino acid at position 6, a set amino acid at position 9, and a set amino acid at position 12, maybe. Okay, and if you don't have these exact amino acids there, it won't bind. So maybe you wouldn't ever have polypeptides in the human genome, let's say, that has that combination of amino acids where you have this specific amino acid here followed 2 steps down by this specific amino acid here, which is then followed 3 steps down by this specific amino acid here, and then another 3 steps down by this amino acid here. Okay, at least that's the attempt. It doesn't work completely. But basically, MHC class 2 molecules, they are not specific for antigen fragments. They just need an antigen fragment to have a few correct anchor residues, and then the amino acids that are in between, they can be whatever you like, basically. So, people who have this HLA-DR4 type of major histocompatibility complex class 2 molecules, they may well present some of the antigen fragments of these citrullonated proteins, which the other forms of MHC class 2 might not. Okay, and that might be what gives them increased risk of developing rheumatoid arthritis. And I should just add that HLA stands for human leukocyte antigen, and it's the old name for major histocompatibility complex. and the genes are still named after human leukocyte antigen. Okay, so MHC, just to add that in, that stands for major histocompatibility complex. Okay, so in the human genome, you have these 3 separate loci which give instructions on how to build MHC class 2. You have an HLA-DR locus, you have an HLA-DP locus, and an HLA-DQ locus, and they all produce slightly different MHC class 2 molecules, okay? Each of them has many different alleles, basically, that you could have, okay? So individuals specifically with HLA-DR4 alleles in this HLA-DR locus on chromosome 6 specifically, it's all on chromosome 6, okay? They are at increased risk, 4 times increased risk of developing rheumatoid arthritis. So major histocompatibility complex, and then it's class 2. Right, okay, so where were we? So, we were discussing the production of these autoantibodies against citrullinated proteins. Okay, so something happens that means that you start producing antibodies, these autoantibodies, which will bind to citrullinated proteins, and this isn't well understood at all, which citrullinated proteins you actually produce antibodies against, and it's not understood really how the failure in immunotolerance occurs. Okay, right. Now, these antibodies, however, have been given a name. They are often referred to as ACPA. Okay, and this stands for that's the first A, citrullinated proteins, that's the CP. Okay, so anti-citrullinated protein, so ACP, and then antibody is the final A. Okay, so you have lots of these ACPAs within your blood, okay? And this occurs years, potentially years. It's up to years, so it might occur only a few months before you actually get rheumatoid arthritis, but it can occur years before you ever get rheumatoid arthritis. So you can get the appearance of these anti-citrullinated protein antibodies within the blood years before you actually get rheumatoid arthritis, and this is what's going to eventually lead to rheumatoid arthritis. Now, for that reason, these anti-citrullinated protein antibodies are also called rheumatoid factor, okay? So, initially, when people found the presence of these anti-citrullinated protein antibodies within the blood, which then led you to develop rheumatoid arthritis, they were called rheumatoid factor, or RF. Okay, right. Now what we need to discuss is the transition phase, okay? So this is the prearticular phase. This is the phase long before the joints become involved. So you've just had this failure in immunological tolerance, and you've produced these antibodies against citrullinated proteins. And you might be wondering, so what does this do? Surely this is going to cause major problems. Why does it take years to get any sort of sign that you've got these anti-citrullinated proteins? Surely they're going to bind to citrullinated proteins and cause major havoc. Well, basically they don't get access to the citrullinated proteins. Now we need to discuss the involvement of the joints, basically. So, in the case of the joints, you do have citrullinated proteins within the joints, and the specific proteins which are citrullinated and are the target for these anti-citrullinated protein antibodies is not understood, is not known even. But we know that there are citrullinated proteins in the joints, and one of these must have an antibody that is targeted against that specific protein once it's been citrullinated. And you might wonder, well, what's the big deal with the citrullination? Well, basically, when you change arginine to citrulline, it does often lead to a huge change in the structure of the protein. So it leads generally to it folding in a totally different way, basically. So that's why you can develop an antibody that binds specifically to the citrullinated form of the protein rather than the non-citrullinated form. Okay, right. So basically, in the joints, in the synovial joints, you have two major forms of this peptidyl arginine deaminase enzyme. You have PAD2 and PAD4, okay? And these must citrullinate, at least this is what the current theory is, that these citrullinate some unknown protein, okay? So some unknown protein in the synovial joints is then converted to a citrulinated protein, okay? And it's still unknown, of course, but it's converted to a citrulinated protein. And I should stress that although these anti-citrulinated protein antibodies are all grouped together as ACPA, of course, they're not just going to bind to any old citrulinated protein, so don't be misguided into thinking that they're just binding to citrulline. No, they're binding to a specific citrulinated protein, okay? So, one of the antibodies that's against a citrulinated protein that you produce must be against some unknown protein that we don't know yet, which is within the synovial joints. And these two enzymes which are present within the synovial joints, peptidyl arginine deaminase 2 and peptidyl arginine deaminase 4, are responsible for converting this into its citrulinated form. And then you've got an antibody that is present within the blood which will bind to this. Okay, so you might think, well, that still doesn't explain why the instant you get these anti-citrulinated protein antibodies being produced, you don't get rheumatoid arthritis. And the reason is that basically the anti-citrulinated protein antibody cannot get to the citrulinated protein in the synovial joint. It's in the blood. The citrulinated protein is in the synovium. It's in, it's outside of the bloodstream. So how is the antibody supposed to get to the antigen? Well, you might say, well, how does it work usually? Usually you produce antibody and it gets into the tissue fluid, but that's because you have an inflammatory response going on at the place where you've got an infection. So usually if you've got a pathogen invading your tissue, okay, you get a massive great inflammatory response. All of the blood vessels, well, all of the capillaries and the post-capillary venules retract their endothelial cells and open up huge great gaps in the side of the blood vessels to allow fluid from the blood to leave the blood and go into the tissue fluid. That's how antibodies usually get out at sites where you've got a pathogen invading, and therefore how they gain access to the pathogen. In this case, there's nothing happening in the joints, okay? So the blood vessels have very low permeability to antibody, so they're not just going to let the antibody get out and go and find the citrulinated protein. Instead, what has to happen is that you have to get some sort of damage to the joint. But before you do that, you also have to have another little transition, okay? And we'll begin discussing the transition phase in the next video.